All right, this is section 7.3, the oxidation of pyruvate and the citric acid cycle. We have two goals. One, explain how a circular pathway, such as the citric acid cycle, fundamentally differs from a linear pathway, such as glycolysis. And two, describe how pyruvate, the product of glycolysis, is prepared for entry into the citric acid cycle. We are going to start with a breakdown of pyruvate, a three-step process. Step 1. A carboxyl group is removed, releasing a molecule of carbon dioxide. This results in a 2-carbon hydroxyethyl group bound to the enzyme. This step is done twice because glycolysis produces 2 pyruvate molecules for each glucose molecule metabolized. Step 2. Hydroxyethyl group is then oxidized to an acetyl group. Electrons are picked up by NAD+, making NADH. The high-energy electrons from NADH will be used to generate ATP. Step 3. The enzyme-bound acetyl group is transferred to coenzyme A, and this produces an acetyl coenzyme A. This acetyl coenzyme A is then converted to carbon dioxide using the citric acid cycle. During the second stage of glucose metabolism, whenever a carbon atom is removed, it is bound to 2 oxygen atoms, producing carbon dioxide, one of the major end products of cellular respiration. The citric acid cycle takes place in the mitochondria. Almost all of the enzymes included in the process are soluble. The cycle is a closed loop, unlike glycolysis, which is a linear pathway. It is a series of redox, dehydration, hydration, and decarboxylation processes. There are 8 steps. Step 1. Coenzyme A is bound to a sulfhydryl group and then eventually combined with another acetyl group. This is a highly exergonic, meaning losing a lot of energy step, and irreversible. The speed and rate of completion of the step is controlled by a negative feedback loop with the levels of ATP. An explanation: if ATP levels decrease, the rate of completion increases. Step 2. Citrate is converted into its isomer, isocitrate. Citrate loses one molecule and gains another. Step 3. Isocitrate is oxidized, producing A-ketoglutarate using a molecule of carbon dioxide and 2 electrons. This reduces NAD+ to NADH and releases carboxyl groups that form carbon dioxide molecules. Oxidation and decarboxylation processes. This is regulated by negative feedback from ATP and NADH and a positive effect of ADP. Step 4. A-ketoglutarate is oxidized to produce a succinyl group. Steps 3 and 4 are both oxidation and decarboxylation steps, which release electrons that reduce NAD+ to NADH and release carboxyl groups that form CO2 molecules. Coenzyme A binds the succinyl group to form succinyl coenzyme A. The enzyme that catalyzes step 4 is regulated by feedback inhibition of ATP, succinyl coenzyme A, and NADH. Step 5. A phosphate group is substituted for coenzyme A, creating a high-energy bond. This energy is used in substrate-level phosphorylation to form GTP and ATP. 2 forms of isoenzymes are used for this step. One is found in heart or skeletal muscle, which is an aerobic environment, to produce ATP, while another type of enzyme is found in the liver, an anabolic environment, in order to produce GTP. Step 6. The conversion of succinate into fumarate. A dehydration process converts succinate into fumarate. 2 hydrogen atoms are transferred to FAD, producing FADH2. Step 7. Water is added to fumarate and malate is produced. Step 8. Oxaloacetate is regenerated by oxidizing malate. Another molecule of NADH is produced. The products of the citric acid cycle All 6 carbon atoms from the original glucose molecule are incorporated into carbon dioxide. Each turn of the cycle produces 3 NADH molecules and 1 FADH2 molecule. These carriers connect with the last portion of aerobic respiration to ATP molecules. To ATP respiration. 1 GTP and 1 ATP are also made in each cycle. The cycle is amphibolic, both catabolic and anabolic.